Welcome everyone to this week's edition of the Extracellular Vesicle Club. So we're going to have a paper today that was just recently published in the Journal of Extracellular Biology, which is of course one of the two journals of the International Society for Extracellular Vesicles, along with the Journal of Extracellular Vesicles. And before we go to our scientific presentation, I just want to remind everybody that if you'd like to join ISEV at the big international annual meeting. It's going to be in Vienna, Austria this year in April. And our early registration deadline is coming up next week. So you have until March 7th to get your registration in. It's going to be an extremely exciting meeting. I've seen a lot of what's happening behind the scenes with the International Organizing Committee. They have a lot of new features this year, and there will also be a lot of new people, people in terms of both the sponsors and the other attendees. So please don't miss out on this meeting. But today I'd like to introduce Joseph Brealey. And Joseph is at NanoFCM. So some of you are using the instrument that NanoFCM makes. Others of you wish that you were using it. I'd just like to stress this is not an industry talk here. This is a scientific talk. This was published in a peer-reviewed journal. And that's the reason that I've invited Joe to come today and talk to us about I think, a very important topic, the topic of fluorescent dyes and how specific they are for EVs, what else they might be labeling, and how we can measure all of that using single particle technologies. So, Joe, thanks again for being here today, and I'd like to hand the screen over to you.
Perfect. Thank you very much for the introduction. Yeah, as Ken said, I'm Joe from NanoFCM. I'm here to talk about a paper we published recently about using membrane dyes to label EVs and assessing how good these membrane dyes are at labeling EVs, how specific and how sensitive they are. Again, as Ken said, a very quick disclosure: I do work for NanoFCM, and this was part of my employment, but it is a piece of research that was properly published. This is our graphical abstract, and again, I think it illustrates quite nicely what I'll be talking about today. So we're looking at EVs isolated from a complex source. So in our case, we're looking at conditioned media and biofluids, so in our case, plasma. And what we're doing is we're isolating these EVs by size or density. In particular, our experiments were all using size exclusion chromatography, so we're isolating by size. And we're hopefully removing all of the majority of the non-EV particles to leave us with these isolated EVs. As we know, though, unfortunately, these isolated EVs will not be pure. They will contain some non-EV particles. And the aim of this research was to investigate membrane labels, so dyes that are able to integrate into the lipid bilayer, and to see if we can find what we would call an ideal membrane dye. And I guess the two characteristics of an ideal dye that I've got here are high efficiency, so it must bind all of the EVs in our isolate, and it's a high specificity, so it must be specific to these EVs without binding to these non-EV particles. A very quick introduction to the technology that we used. It's nanoflow cytometry. It's very, very similar to regular flow cytometry, where we're passing extracellular vesicles through lasers to get information on their size, concentration, and fluorescence. The outputs that we're going to be looking at today are these dot plots, very similar to flow cytometry dot plots, where we've got the side scatter of our EVs. So we're detecting our EVs by side scatter, and we're able to look at their fluorescence. So I mean, in this example, we've detected about 2,900 EVs, and we see that 1,100 of them are fluorescent, so 40% fluorescent in this case. So why do we want to look into membrane dyes and kind of labeling EVs a bit more generically? Well, one of the reasons is that protein labeling isn't going to hit all of your EVs necessarily. We found a lot using our technology, and especially in the last few years, we're seeing massive variation in the expression of CD9, 63, and 81 between different cell lines, between different sources, and between even different isolation methods. So we have here a bit of a quick example with two sets of EVs derived from biofluid. Not biofluid, sorry, they're derived from cell culture of SW620 and SW480 colorectal cancer cells. And we see that in our SW620 cells in blue, if we stain with a cocktail of CD9, 63, and 81 antibodies, we see about 60% positivity. So about 60% of the particles in this EV isolate express one of the tetraspanins. And the expression of CD63 and CD81 are quite low, both below 20% of the particles. When we look at SW480 derived EVs, it's even lower. We see about 30% of the particles express one of the tetraspanins, and in particular, CD81 is almost absent from this cell line. We then have the other problem that I discussed is that in an EV isolate, it will not necessarily be pure. There are other particles that can co-isolate with EVs depending on whether we're looking at density or size-based isolation. In this paper, we're looking at these small EVs, so we're kind of focusing in on that kind of 40 to 150 nanometer size range. And the primary co-isolate we would expect here is VLDLs. They're a very, very similar size, and it's difficult to separate them from EVs using size exclusion chromatography. When we're isolating membrane dyes, what are we looking for? What makes a good membrane dye? Well, first, it must be sensitive, so it must label all of the EVs in the mixture. It must be specific, so it must not label any non-EV particles. In this experiment or in this research, we used VLDL, so very low density lipoproteins, as a stand-in for a non-EV particle. It must not aggregate, so the dye molecules must not aggregate to form particles that could be detected as like false positive EVs. The dye must label EVs brightly enough that they are detectable using a fluorescence-based system. And ideally, the staining protocol should be quite easy. It should be relatively easy for a new user to pick up this membrane dye, bring it into their lab, and start staining EVs with it. So we got our EVs for this experiment from cell culture. So first of all, we cultured the SW480 and SW620 cell lines and extracted EVs using size exclusion chromatography. This was done by the Nick Peak lab at Sheffield Hallam University. And we isolated EVs from human plasma, again using size exclusion chromatography, and this was done by the Ryan Pink lab for us at Oxford Brookes University. Some characterization of our EVs before we get onto the membrane staining. So we looked at the SW620 and the SW480 cell culture EVs, and we looked at, in A, the protein content of all of our fractions. In B, we've got the size distribution and concentration of our particles. In C, we've got that the staining results for CD96381. I showed those earlier. In D, we've got a Western blot, and E, we've got a TEM image. So with these EVs, what we're going to be doing is staining them with this membrane dye, following a very simple kind of a simple, easy, robust protocol that we developed at NanoFCM. So to begin with, we simply measure the particles unstained to measure their size and particle concentration. We'll then dilute the EVs down to a standard concentration. So we use 2 × 10⁻¹0 particles per mil. To that, we add our membrane dye. We then incubate for 30 minutes at room temperature in the dark. We then do a further dilution step, and then we measure the stained particles. And from this stained particles measurement, we're looking at the size of the EVs, we're looking at their concentration and the staining efficiency. So what percentage of the EVs have been labeled by the dye. We looked at 6 dyes in this paper, so 6 commercially available dyes that claim to be membrane specific. We looked at these dyes called the Memglow. Memglow 488 is green and Memglow 640 is red. They're from a company called Cytoskeleton. We looked at the Exobright dyes where 490, 515 is green and 640, 660 is red from a company called Biotium. And then 2 dyes from Thermo Fisher called Cell Tracker Deep Red, which we're going to refer to as CTDR, and Cell Mask Deep Red, which we're going to refer to as CMDR. And both of those, as you might expect, are red. The first thing that we did when staining our EVs was to work out the correct concentration of the dye to use. So as I say, we use a standard concentration of EVs, that's 2 × 10⁻¹0 particles per mil. And we need to work out what concentration of dye gives us enough dye to label as many EVs as possible, but not so much dye that there's a really high excess concentration and we see a very high background fluorescence. This is our optimization graph for Cell Mask Deep Red. If we begin by looking at the blue line and the left y-axis, we're looking at the fluorescence positivity. So what percentage of the EVs were labeled positive for Cell Mask Deep Red? And as if we look at the high concentrations, the kind of 50 and the 25 micrograms per mil, we see a very, very low proportion. Under 20% of the EVs are being labeled positive. As we decrease that concentration, contrary to what we might expect, as we're adding less dye, we're decreasing that concentration, the percentage of EVs that are being labeled is increasing up to near 100% once we hit 2.5 micrograms per mil. Now, the reason for that is if we look in the orange line and the right y-axis, we're looking at our fluorescent background. So at those high concentrations, the 50 and the 25 micrograms per mil, we have a very high, upwards of 500 and 1,000 fluorescent background. For context, we want this background to be below 50 at all times. So when that background shoots up to like 500 and 1,000, what's actually happening is we can't see any of the stained particles. There's so much fluorophore in the system that we can't pick out any of the stained particles. But once we start decreasing the concentration of dye, as we might expect, that fluorescent background drops because there's less kind of excess unbound fluorophore. And eventually we get to a level where it has a nice low background and quite a high fluorescence positivity. If we then compare this to another dye like Memglow, according to the manufacturer, Memglow, any unbound Memglow will self-quench. So if we add too much Memglow to the system, it's non-fluorescent and it doesn't impact the background. So we see across this quite wide range of concentrations that we've used, a very high staining efficiency and a very low background across the board. And this is kind of one of my points when it comes to ease of use. So a dye like Memglow would be quite, in theory, quite simple for another EV researcher to pick up Memglow, bring it into their lab, and it has this nice wide range of concentrations where it gives you this good background and this high staining efficiency. But if you compare that to Cell Mask Deep Red, with Cell Mask Deep Red, you need to make sure you're doing this really thorough optimization to ensure that you're not adding too much or too little of the dye. When we look at our staining results, if we start with the EVs from cell culture, so these are our SW620 EVs in blue and our SW480 EVs in orange, we can see that 3 of our dyes, so Cell Mask Deep Red, Memglow 640, and Memglow 488, bind very effectively. There's almost 100% of the particles are being labeled by these dyes. When we look at Cell Tracker Deep Red, it's a little bit lower, so 60 to 70% of the particles are being labeled. And when we get to Exobrights, it's lower still. One of the very interesting results that one of the most interesting things I find about this is with the Exobright, we're seeing a very large difference between the two cell types. I mean, if we look at Exobright 640, 660, in the SW620 derived EVs in blue, it's about 20% of the particles, and in orange, it's about 70% of the particles. And what's actually happening here, we think, is that Exobright 640, 660 is instead of binding to the lipid bilayer itself, it's binding to a specific protein on the EV surface. So I think what we're seeing here is very simply that one of our cell lines is expressing that protein more highly than the other. So the SW620 cell line doesn't appear to be expressing that protein as highly. But I think this really, it kind of highlights when we're using these membrane dyes, we need to be sure that we're using one that has a good mechanism of action. I mean, Exobright is protein specific at the end of the day, whereas the things like Cell Mask Deep Red and Memglow bind to the lipid bilayer itself. And we can see some nice dot plot images here with Memglow 488, whether it's the SW620 EVs on the left in I or the SW480 EVs on the right in II. The vast majority of these particles are being labeled positive. There's a quite small population remaining in the negatives. Comparing that again to Exobright 640, 660, on the left in the SW620 EVs, the vast majority, almost 70% or almost 80% of the EVs or 80% of the particles are unlabeled. Whereas in II, we see it's the other way around. We see about 70% labeling. What we then did with our SW620 EVs is we combined membrane labeling with simultaneous tetraspanin labeling. So we stained our EVs with a cocktail of CD9, CD63, and CD81 antibodies, and then simultaneously with our membrane dye. And what we're looking at on this graph is if we just consider those particles which are tetraspanin positive, what percentage of those particles were bound by the membrane dye? So in theory, we should see 100%. These are particles that are tetraspanin positive. They express the tetraspanin, so they should have a membrane. And we see that Memglow, Cell Mask Deep Red, and the Memglow Green do bind nearly 100% of these particles. Now, take that with a pinch of salt because they also bind 100% of the particles that aren't tetraspanin positive, but it's important that we're not missing any of these. When we look at Cell Tracker Deep Red, it's again about 70%. So of the particles that are tetraspanin positive, only 70% of them are being labeled as membrane positive with Cell Tracker Deep Red. And with the two exobright dyes, it's even lower. It's 50% of the tetraspanin positive particles are exobright positive. We can look at the size distributions of these populations as well. So in blue on this middle graph, I have the dual positive population, that is tetraspanin positive and membrane positive. And in orange, we have the population that is tetraspanin positive and membrane negative. So we could consider the orange population as the population that the membrane dyes are missing. So these are particles that have the tetraspanins, they're tetraspanin positive, but they are not being labeled for the membrane. And we can see there's quite a big size difference between them. The blue population is around 150 diameter, 150 nanometers diameter median, whereas the orange population is more like 60 or 70. And we can see it quite nicely on this size distribution graph as well for exobright 640, 660. The orange population is quite clustered around between about 60 and 100 nanometers diameter, whereas that blue population stretches all the way up to about 200. What I think we're actually seeing here, which we'll get to in a minute when we talk about brightness, I think what we're seeing is that these dyes aren't bright enough to label the smaller particles. We're seeing a smaller number of dye molecules bind to each particle, and they're not bright enough to allow detection of these particles. But looking quickly at specificity, so as I say, we use purified VLDLs as a stand-in for a non-EV particle. And again, we stained them with the membrane dye following the same protocol. And we see that the exobright dyes are very good at not binding these VLDLs. There's only about 5% binding on the exobright. With cell mass deep red, it's still under 10%. I think it's about 8%. Cell tracker deep red, you're getting up to about 15% binding on these VLDLs. But the kind of the most important result on this graph is these two memglows, memglow 640 and memglow 488. What we're seeing is that they bind almost all the VLDLs. So they are not specific to EVs. They will bind other lipid-based structures. So again here, yeah, we have some dot plots showing with the exobright and cell tracker deep red that a very small proportion of these VLDLs are in this positive population. They're mostly left unlabeled. Comparing that to the two memglows, again, we see that the vast majority are being fluorescently labeled. So these dyes aren't specific to EVs, and they aren't specific even to a bilayer. They're able to bind these lipoproteins. Staining our plasma-derived particles. So again, we have particles derived from human plasma using size exclusion chromatography. What we see is there's almost two groups here, right? We have cell mask deep red and the two memglows binding a very high proportion of the particles, over 80%, up to almost 100%. And we have our other three dyes, exobright, cell tracker, and exobright green, with a very low, under 20%. I find these results quite interesting. One of the major co-isolates we would expect here, because it's a size-based isolation from plasma, is VLDLs. And if we look at the results here of EV staining, VLDL staining, and plasma staining, on our two memglows in blue, they're very effective at binding our EV preparations. They're very effective at binding VLDLs in orange. So perhaps it's unsurprising that they bind the vast majority of the particles in the plasma. When we look at cell mask deep red, so far our results say that they're very effective at binding our kind of cell culture EV prep, but they are not cross-reactive, I guess, with VLDLs. And yet we're still seeing that labeling of the vast majority of plasma-derived particles. And I think that the main thing this shows us is that we really need to be testing it on a much wider variety of environments and a wide variety of non-EV particles, because yes, cell mass deep red is specific enough to not bind to VLDLs, but there is something else going on in this plasma. It's possibly binding to another component of the plasma. Another key factor of these membrane dyes is aggregation. So we want to make sure that when we incubate this dye in PBS without the presence of EVs, so particle-free PBS, we want to make sure that these particles don't form kind of EV-sized micelles that could be detected as false positives. And we see that we do see that across the board of these 6 dyes we tested, we see a very low concentration of aggregation. There's some dot plots here for exobright, cell tracker, and memglow green, and we see, yeah, just a fairly low concentration of particles here, not too much to worry about. What I'm comparing this to is a lipophilic dye called PKH67, which I think is fairly commonly used for EV staining. And what we see with PKH67, it didn't make it into the paper, unfortunately, but what we see with PKH67 is when we incubate it without EVs, just in its binding buffer called diluent C, we see a very, very high concentration of these EV-sized micelles. We're seeing micelles in this kind of 50 to 120 nanometer range, the vast majority of which are fluorescent. So when we stain EVs using our system with PKH67, what we end up with is we can't tell the difference between these PKH micelles and true PKH stained EVs. It's a very big thing to be aware of. Looking at the brightness of our EVs, I've quantified the brightness here by using the stain index, which is effectively how much brighter are the stained particles than the unstained particles. And it's basically just a measure of how bright, almost like a signal to noise ratio. And we see that Memglow 488 is far and away the brightest of the bunch, with the stain index around kind of 250. I do find it sometimes quite hard to interpret these graphs though in the numbers, so I've got some very nice pictures here. This is what we observe when we measure EVs stained with Memglow 488. As the EVs are passing through our laser, we see this peak, this peak of fluorescence. And we can see that the majority of these peaks exceed the y-axis here, which is 500. So the majority of the peaks here are over 500. They're very brightly stained EVs. If we then compare this to Exobright 49515, the brightest event we can see here is about 350. So with our Exobright stained EVs, they are across the board a lot dimmer and a lot harder to detect. And I think this is why we seem to be, quote unquote, missing some EVs, particularly when we looked at the tetraspanin positive particles. I think what's happening is that the smaller particles just have fewer dye molecules, and the dye molecules themselves aren't bright enough to allow detection of these very smallest EVs. So in summary of the parameters we looked at, we can see that the Memglow dyes at the top come out as fairly promising candidates. Like, they're very sensitive. They bind the vast majority of EVs. They are quite bright, so we can see even these small EVs. There's no bias towards just looking at the larger ones, and they don't aggregate in their working buffer. But the big drawback to the Memglow dyes is that they are not specific to EVs in any way. They're very good at inserting into any kind of lipid-based structure. Obviously, here we tested them on just VLDLs, but since we've tested them on kind of a much wider variety of lipid-based particles, and we see they just bind to everything. Which, as long as we know that, they are quite a useful tool. The Exobright dyes they seem to be very specific. They didn't bind any of our VLDLs, and I haven't observed them to bind any other kind of non-EV particles. They don't aggregate in their buffer, but they're not very bright, right? They don't allow detection of these very smallest EVs. So we're losing that sensitivity, and we're introducing a bias towards just looking at the larger end of this size range. When we look at cell tracker deep red, again, it's very sensitive. It seems to bind the majority of particles. It doesn't aggregate, but again, it suffers with that brightness. It suffers with introducing a bias towards larger particles, and it does suffer to an extent with specificity. Cell mask deep red, again, emerges as quite a promising candidate. It's very sensitive. It binds, again, almost 100% of EVs. It doesn't aggregate in its working buffer, and it is... Specificity here is based on our VLDL binding. I think given the plasma binding, I'd probably downgrade that to a yellow. I think we need to widen the net there on specificity, but it's also not incredibly bright. But it does seem to be a better option if there is kind of a VLDL or lipoprotein contamination. Although another thing to consider is that didn't make it onto this table, but is the ease of use. As I showed at the beginning, the Memglow dyes have a very wide range of concentrations that they are kind of applicable at. There's a wide range of concentration where we get very good staining and a very low background. Whereas with cell masks deep red, you have to perform that very sensitive optimization to make sure that you're hitting the right concentration so that you don't introduce a fluorescent background. I think the real conclusion is that none of these 6 dyes that we tested is the quote unquote ideal membrane dye. They all have situations where they are good, and they have situations that introduce quite big drawbacks. So what are we going to do about it? What are the next steps for this project? Well, our main next step is to test more dyes. There's new membrane dye products going onto the market seemingly every day. So there's a lot more work out there to try and find this ideal membrane dye. We'd like to look as well at the transfer from stained to unstained particles. So if we have two populations of particles where one is membrane stained and one isn't, and we mix them together, do we observe that the dye kind of leaches from the stained population to the unstained population? We've also talked about looking at fluorophore bleaching. So I wonder if some of the brightness issues we're seeing is because the fluorophores bleach much more quickly than the others, and we need to be much more careful to isolate them from the light. And of course, we used VLDLs in this study as a non-EV particle, but I think trying to stain other particles that can commonly co-isolate in EVs from different biofluids in different environments is always going to be valuable work. And I guess just to finish off, I'd like to thank my colleagues and some ex-colleagues at NanoFCM for helping me out with this. And then the Nick Peake lab at Sheffield Hallam University and the Ryan Pink lab at Oxford Brookes University for A, providing the EVs, and B, just being brilliant help throughout the paper writing process. So thank you. I think I've finished a little bit earlier than planned, but I am more than happy to answer any questions that anyone has. Thank you.
Thanks, Joe. Yeah, that you really went through that, and I think there are a lot of questions for you. I have a few as well. I think I'm just going to start with just noting that I think your results demonstrate that the choice of dye is going to be dependent on what substrate we actually have, right? And if you have an extremely pure population of EVs, well, then maybe one dye would be better than another. Whereas if we have this background that we know about, there might be some difference. And on that note, you measured aggregation in PBS, and you showed that the PKH dyes are just aggregating like nobody's business. What about aggregation in a more complex medium, such as plasma that has been depleted of all particles, larger than whatever nanometers? Did you do any experiments like that, or do you have any plans to do that?
It's not something we tested, but it was one of our ideas with the cell mask deep red result. We see this very high staining percentage in plasma. One of our thoughts was aggregation. It's something we'd like to look into, definitely.
Oh, and Melanie, I apologize. I see that you just asked this question too. If you have a follow-up, please, actually, I've got to go in and allow people to unmute themselves here. Just one second. All right, so I've allowed now the unmuting, and I'll just call on people. Melanie, did you have a follow-up to that?
Hi, sorry. Yeah, I think it's really the knowing how stable they are is really important to follow them. So yeah, I think, I don't know what the experiences of others are for lipid dyes. Hi, sorry. Yeah, I would be really interested to see that.
It definitely is absolutely something we want to look at. Yeah, we found some very, very interesting results. I mean, obviously, take Memglow, for example, that binds a very, very high percentage of VLDLs and kind of other lipid particles. That means we have to be very careful using it if there's going to be other lipid contamination. But we've actually found it very useful for staining things like lipid nanoparticles that are synthetic, and you want to stain every particle in there, and you don't expect there to be other contamination.
Yeah, thanks.
Maybe also just a brief follow-up on that. What do you consider to be the limit of detection in terms of sizes of particles that you can measure?
Sure. So with the side scatter that we were using, we were looking down to about 45 nanometers. So the range we were looking at was 45 to 200 nanometers. With I mean, again, I'm coming back to Memglow. It is so bright that we can detect fluorescent events below that limit, which I think, according to the tests we've been able to do, I think are EVs below that limit. So the use of a very bright dye can extend this limit of detection below what the instrument allows.
Good. All right. Okay, I want to go
May
to
I ask something
go
on
ahead.
these limits? Do you know what is the number of fluorophores you need to be able to detect something, or do you have any sense of that? How many of these Memglow?
Absolutely. So For
have to be on a particle? Yeah.
For memglow specifically, I don't. Obviously, I can only speak for our instrument, the nano analyzer. We tend to say it's somewhere around 3 to 6 fluorophores if it's bright enough. But again, it's hugely dependent on the fluorophore.
Okay, yeah. I think we looked a lot into fluorescent imaging and also wanted to find out what is our limit. And for the bright ones, of course, there are very many different ones. We also feel this is somehow like 5 to 10 fluorophores is the limit. Yeah. But it's good to see what with different instrumentation or techniques can be achieved. Thanks.
No problem.
I'd like to call on Devika next. You have a technical question, but also some suggestions. Go ahead.
Thank you. Thank you for a great talk, Joe. So my first question is technical on the detection limits of NFCM. I think I just heard you say that it can go from 45 to 200 nanometers. Is that right? So would you say 45 with an upper limit of 200 nanometers?
Yeah, so within using the nano analyzer, it's 45 to 1,000, and we have a couple of different laser settings that we can use to more specifically look at particles. So in this experiment, we were using a kind of specific laser setting just to look at 45 to 200.
Thank you. That's very helpful. So my question was, so when you think of, up to 1,000 nanometers, do you think that nano FCM can reliably distinguish small versus larger vesicles just based on changes in their side scatter?
Yeah, definitely. I mean, we've got some really nice data. Obviously, this didn't make it into this presentation, but we have some really nice data available that shows that we do see very accurately sizes between 40 to 1,000. So we can tell the difference between these kind of small EVs in the I mean, like I say, 40 to 200 nanometer range, comparing to these large EVs that are between 200 and 1,000.
So if I can ask one more follow-up to that. So I will read more upon your work to see. I don't know if that maybe we can talk more offline. But so if nano FCM is distinguishing sizes based on side scatter granularity, does the granularity vary considerably for a small EV versus a large EV?
Good question. So we are looking at sizing using this side scatter intensity relative to a silica nanoparticle standard. So it's all to do again with that refractive index. Across our tests, we've seen good results from the whole range from 40 to 1,000. So it doesn't seem, the refractive index doesn't seem to change much in this range.
Thank you. That's interesting. And I have one more quick question. So have you thought about using something like calcine acetoxymethyl ester, because it's a dye that is cleaved by esterases, so it's kind of it's kind of not orthogonal, but it does not rely on lipophilic interactions to label EVs. Have you any thoughts on that?
Absolutely, yes. So we used CFSE, which I think is very similar. It's a very similar mechanism of action. And again, that's as you say, dependent on esterase activity. So it's more looking at intact functional EVs as opposed to just looking at the lipid bilayer. And we got some interesting results, but I think for whatever reason the cell lines we were using weren't particularly good for staining with CFSE. They seem to have maybe quite a low enzyme expression. We got fairly poor results on our 2 cell lines.
Thank you. That was a great talk. Thank you so much.
No problem.
Thanks for the questions. Next is Andrea.
Hi, thank you. I was wondering if you have the same data that you have shown for PKH regarding the aggregation for dyes like the IR and the family and dyes of the same family, since they are so much used in vivo. This is particularly relevant that doesn't happen.
So we tested PKH 67 and PKH 26, and we saw those kind of a very high concentration of aggregates. We didn't look at any of the DI series, but it's future work is more dyes, so we very well might in the future.
Okay, thank you.
And Christina.
Hi there, beautiful talk. So Devika asked some of part of the question in terms of labeling inside EVs instead of the surface. So you shared that your enzyme didn't seem to, it didn't seem to have as much enzyme. So that's something obviously that we also need to consider. How do you account for lipoproteins that are associated directly or attached to to the EVs themselves? So you, instead of thinking of them as separate, so the reason I ask this is I did some work where we took literally an EV prep, and I had someone do an LDL HDL isolation, and she was able to get like lipoproteins from the EV prep. It was a dirty precipitation, but at the same time, what if they're not distinct populations and they're co-associating? Like, how do we, or is that something that you think you could tweak apart using this type of work?
Really interesting. I guess if the kind of EVs and the VLDLs were attached and kind of we would see them as one particle, and if our membrane dye labeled the lipid bilayer, I guess it would be indistinguishable. I guess we could look a bit more at the size of these particles, because they would probably come through as double the size. Maybe we'd see two populations. We'd have maybe an EV population and then I don't know, EV plus VLDL kind of aggregate population. It's a really interesting question though, but
yeah, because it seems like your MemGlow, which stained the best, also stains VLDLs, right?
Yes.
And so are they associated with the EVs directly, or yeah?
It's a great question, and it's one of the kind of roadblocks that we came up against in this whole study was we have these EV preps that we're going to stain with our membrane dyes, but should we expect to see 100%, or should we expect that there will be some contaminants in here? And it was a question that I'm not sure we've been able to answer yet.
Yeah, because the thing and maybe you can tell me if this is voodoo, but I am always concerned about when we label the surface of an exosome is that when it's taken up by a cell, for example, you get fusion of membranes and you lose the dye. Like, how do we allow for that if we're not staining the inside? So I've always preferentially gone for staining the inside, but that could just be some sort of voodoo-ness in my thinking. Do you have any thoughts on that?
I think it's definitely good thinking. I would say like almost the ideal situation would be if we could label every EV with a dye like CFSE, but then you're kind of relying on the assumption that every EV contains some of these esterase enzymes, and it's all going to be properly labeled. It's a great question.
No, thank you. Your talk was very thought-provoking. Thank you.
Thank you.
Thanks for the questions. Next is Ming Lin, a question about the VLDL. Fascinating question.
Thank you very much for your talk. Very good. Try to address a lot of technical issues. And here the question is, have you tried to look if the isolated VLDL combined the antibody against the tetraspanings? As you see the dead cell membranes may be picked up by the VLDL when it's circulating in the circulation.
That's a great point. It's not something I thought to do, but I mean, I would hope that none of the VLDLs would bind the tetraspanin antibodies, but it's a great point.
Yeah, many membrane markers, when they die, they may, circulate in the circulation, and the VLDL, catch all the kind of lipid. So that may be, good to have a look. Thank you.
That's fascinating, because then a VLDL that would have some of that EV marker might very well be mistaken for an EV.
Yes, thank you.
Thanks. Next question is from Maria S. So the question is, red fluorophores in general tend to be dimmer, so perhaps a cell mask green instead of red. Is that something that you contemplate testing?
Great point. It's certainly what we observed anyway, that the red fluorophores in general were a bit dimmer. That there is a cell mask green available, not something we tested, but again, it is on the list.
Next question is a MR Agotha. How to find the fluorescence aggregation using NanoFCM. So you're just looking at, you're just introducing the dyes, correct, into a buffer, and then you would detect those particles, correct?
Absolutely. So we typically stain 9 microliters of our particles, but yeah, we take a certain volume of your buffer, so PBS, TE, whatever buffer you might be using, introduce to that the same concentration of dye that you're using to stain your EVs. Leave it for, we say 30 minutes, but somewhere between 15 to 60 minutes, and just measure that in the system and see what you see. And what we're hoping to see is almost nothing. What we're hoping to see is that there's no events introduced by doing this.
Good. All right. Next question is from Kaylin. Kaylin, I think you really want to put Joe on the spot here. Go ahead.
Wonderful presentation, Joe. I am actually about to start a study using the nano FCM, kind of more of like a characterization method after doing isolation using a different assay. And so I wanted to see what dyes would you actually recommend when using plasma-derived particles, since we're specifically looking at the EV presence in that area.
Brilliant question. And I think based on the results that we got in this round of testing, I think that it is slightly unclear. I mean, I definitely wouldn't recommend either of the memglows for use in plasma, because they are just going to bind to every kind of lipidic particle in the system. I'd be tempted to suggest cell tracker deep red. I mean, it labeled quite a high proportion of our EVs. When we looked at the tetraspanin positive particles, it labeled 70+% of them, and it didn't seem to have this reaction in plasma where it stained every single particle. So I would be tempted to say cell tracker deep red from this selection.
So maybe I will read Johan's question here. How would you standardize each of these fluorescent dyes in terms of the MESF or ERF units?
Absolutely. So that is one of the things we've been working on in the last year, is moving towards this more quantifiable, these quantifiable measurements using MESF and ERF. So, I mean, we've got sets of fluorescent beads, and you can buy fluorescent beads, for flow cytometers and things like that, which have are quantified in terms of MESF or ERF. And then I guess you'd have to look at the fluorophore on the membrane dye and maybe look at its optical properties and try and develop a correction factor to convert the maybe MESF or ERF of a more kind of standardized fluorophore like FITSI or one of the Alexa fluorophores, maybe to the fluorophore in the dye. But yeah, it would just be using a set of standard beads and looking at the fluorescence intensity relative to them.
Next question is from Mahmoud. Mahmoud, do you want to ask this?
Yes, thank you very much, Joseph, for this presentation, and thank you for the organizer for sure. I just I'm agreeing on using, for example, dyes that are going to stain something inside. I'm agreeing on the point in which that it doesn't make sense, for example, because for me, for sure, I work with bacteria. And whenever you change the conditions of the environment, you have different kind of or types of EVs that not all of them for sure will have esterases, and not all of them will work in the same manner. However, I don't think that there is an optimal dye in which you want to like stain something outside. It depends on the condition or the environments that you are working with, I think so. But I just want to know some technical information from you. Like, for example, did you perform any or try to perform any purification techniques to reduce the background or maybe non-specificity problems? This is the first question. The second one is like, did you by chance during the different analysis of your EVs, did you have problems with the stability of the EVs itself, or and how you can differentiate between the stability of the isolated EVs and the efficiency of the labeling dye, like how to differentiate between both of them? Because I know, for example, different buffer that you resuspend your or you use to resuspend your EVs, depending on the isolation technique, can basically affect the viability of your EVs. So, for example, PPS, if you use it, it can decrease the stability of EVs, and you cannot store it for a longer time and reuse it again. So, during your analysis, for example, or during doing, I don't know, a nano-tracking analysis or whatever, how you can distinguish between the stability of the EVs and the specific and the efficacy of the dye? Thank you.
Definitely. So I guess in answer to the first question about purification to maybe remove a fluorescent background, with our optimization series, with our kind of dilution series, we're making sure that we add the correct amount of dye that can stain all of the particles without leaving any too much unbound dye. So we tend to not need to do any further purification, but in cases where we do need to do it kind of more generally, we will usually use ultracentrifugation or size exclusion chromatography. Regarding kind of the stability of the EVs, so we always make sure that when we're doing the staining, the EVs are kind of in the PBS and at room temperature for as short as possible. It's only half an hour, and we always make sure to use a control that's just EVs plus 1 microliter of buffer rather than the dye. And we didn't struggle too much. I mean, as I say, they're only at room temperature for half an hour. We didn't see too much degradation while we were staining the EVs.
Okay, thank you so much.
No problem.
Thanks for the question. Next question is from Kieran.
Hi, thank you for your talk. I was just curious with the cell tracker dye, since it's esterase dependent. Were you labeling the EVs or the EVs and VLDL after purification or before? I know you have to clean it up to remove the dye afterwards, but because there's soluble esterases, would you expect that there might be some esterase reaction with proteins outside the EVs, or like say coating the surface of lipoproteins? That might be a reason why your VLDL might be picking up some label, that there might be dye being broken down by soluble esterases that might be coating the surface of VLDL particles.
That's a really, really great question that I kind of didn't think of while we were doing this. I would say the VLDLs that we used in this were purchased from Thermo Fisher. They're not something that we isolated ourselves, so I assume they're highly pure. But it's a great point. Yeah, if there are soluble esterases, they'll likely be breaking down the dye molecules, and it could allow them to bind, non-specifically.
Yeah, another, I think, a really good point. We got an LDL prep commercially, and when we looked in that LDL prep, even by EM, there were definitely some EVs in there and probably some other things too. It was very pure, but but very pure sometimes is not pure enough. And certainly I have seen data where CFSE is reacting with something outside of EVs. So all very good points. Olivier. See, Olivia asks, what would be the minimum EV input for accurate detection? So what do you go for when you're trying to find your right dilution?
So again, obviously I can only speak for our instrument, the nanoanalyzer, but what we do in our protocols is we dilute the EVs down to 2 × 10⁻10 particles per mil, and then we dilute them around 100-fold for measuring in the instrument. So the concentration that actually goes into the instrument is about 2 × 10⁸ particles per mil.
To the 8th, you said?
Yes, 2 × 10⁸ particles per mil.
Our next question, I think that Andrea asked this question, but there's a follow-up. I would be similarly interested in TFP dye analysis. So here's another one maybe to add to the list.
Yeah, the ever-growing list of new membrane dyes, but definitely it's all stuff we want to look into.
Yeah, thanks for the suggestion, Sierra. Let's see here now. Morali, you have a question about antibody concentration.
Hi. So my question is, when we are using 10⁻10 particles with the tetraspine antibodies like CD63, CD9, or CD81, what's the antibody concentration should be used for the particles?
Great question, and I'm afraid the answer is going to be it's dependent on how how effective the antibody is at binding the target and how bright the fluorophore is, I would just recommend taking those EVs down to a standardized concentration and staining with a dilution series of the antibody. And we want to be adding enough antibody to saturate the targets, but not so much that it introduces a fluorescent background.
Thank you.
And Johan Kim has another question about post-labeling.
Yeah, so
Go ahead,
sorry,
Johan.
yeah, great talk, Joseph. Also, I just missed the opportunity to ask you a previous question, but so I found out sometimes those free fluorophore, even though you said like 100%, almost close to 100% labeling efficiency, but have you ever tried any like those labeled EV with column, try to see whether like those the fluorophore bound to correctly to the extracellular vesicles, or it was to see if it was kind of some, you said micelles, the presence of micelle still like left over in the sample? Thank you.
It's not something we did for this study, but I have done some, post labeling particle isolation recently, and it can be really helpful. I mean, in cases where maybe the dye has quite a low binding efficiency, so you have to add a lot of dye to stain the particles, and that gives you a fluorescent background. Being able to isolate away that background and just being left with the particles is very helpful. And it is a good point, like having these maybe micelles that can be detected as false positive UV particles. That's really what we're testing for when we're looking for aggregation. So I don't think it's too much of a problem with these dyes, but it's definitely like if we were seeing maybe a much higher labeling efficiency than we would expect, it could be something to look into to make sure we're purifying these particles and not seeing any false positives from the dye.
Great, thank you.
Thanks for the question. Now we have two questions from Hui Chao.
Hello.
Hi there.
Can you hear me?
Yes, we can. Go ahead.
I have two questions. Have you ever tried samples from bacteria, the EV samples from bacteria?
So I've personally never worked with bacterial EVs, but I know I've got some colleagues that have, and it's just a case about using specific markers and making sure we test membrane dyes on samples first.
Yeah, according to my experience, so I prefer some hydrophobic dye, which is nail red. It's only fluorescence, the hydrophobic environment, which can exclude much background of like non-specific fluorescence because it's only have attached to the hydrophobic one. But some dyes like I forgot, some green dyes, it's fluorescence even in the buffer. There's lots of background. I think I'd suggest you use some like hydrophobic dyes. But again, this is like the specific is another problem. Sometimes you have to concentrate your samples a lot. That's a lot of problem.
Definitely. I think that's one of the big things we were seeing, like the differences between like cell mask deep red, which is always fluorescent, it's just bound to a fluorophore, comparing that to memglow, which is fluorescence is activated upon it binding to the lipid bilayer. So it has that really nice low background. It's definitely a lot nicer to work with, a lot easier to work with.
Yeah, that's my experience. The other thing is about like standardization. When you pick up like samples from different source, maybe you use like you said, okay, this is the standards are diluted 10 to 9, but up to which equipment you use, it may not the pure the vesicles, it may contain some other particles. How can you exclude this possibility?
Absolutely, and that's kind of one of the limitations here, is that we're looking at particles, right? And we have our EV isolation, and when we're staining that with membrane dyes, we see 100% labeling. And the question is that real? Are those really 100% EVs, or are there other particles in there that we're seeing non-specific labeling? So should we expect maybe to see maybe more like 70%? And it's a very difficult question to answer because again, we can't seemingly we can't rely on this tetraspanin labeling because not all of our particles are expressing one of the tetraspanins. Yes, a very difficult question that we would like to answer ourselves
Yeah.
of how can we be sure?
Mm-hmm. Yeah, thank you. Very good, helpful.
Very good, thank you. And on the topic also, I think of the, let's see, where is this? The hydrophobicity, Pascal. We can hear you.
Okay.
There you are. Hello.
So
Hey, Pascal.
we just starting playing with nano cytometry or flow cytometry, how to say. So I was wondering whether you, when staining tetraspanins also with directly labeled antibodies, did you observe more non-specific signals with Alexa 647 dyes than with Alexa 488, for example, because 647 being considered quite hydrophobic, I was wondering whether it's okay for nanoflow cytometry.
Good question. So, I mean, in this study, we didn't use any kind of Alexa flow conjugated antibodies, but we have done some really nice, I say really nice, I was the one that did the work, but I think it was really nice work recently, staining EVs using the same antibody conjugated to Alexa flow 647 and Alexa flow 488. And what we do see is very similar results. We see that the fluorophore choice in this case with these antibodies doesn't seem to matter too much. They are both suitable.
And then I was also wondering, did you test the EVs from knockout cells as negative control as well? So, for example, if you look at CD9, did you test EVs from CD9 knockout cells as negative control? And
no, it's not something we looked at, but again, it's a great point.
okay, thanks.
Thanks, Pascal. And I think in the interest of time, I'm just going to note that there's been a nice discussion here about the preservation of EVs and, at different temperatures. Maria is asking if you think that the particles that were positive for the tetraspanins are intact or if they could be membrane fragments. And there's also some discussion here about maybe using trehalose as a cryopreservative and whether that needs to be included if you're just keeping samples at 4 degrees Celsius. So I think a lot of variables that can go into the sample pre-processing and before you even put something on the instrument. Are those preservatives and anything that you're looking into, Joe?
I mean, again, these were kind of prepared by other labs with our collaborators, but as far as I know, no. As far as I know, there's no kind of preservatives in there.
Okay, well then we have come to the end of the questions. And I'd just like to thank you, Joe, for being here today and for sharing your very exciting work. The number of questions that we've had, I think, speaks to how interesting the community is finding this, because we all want to be able to label and track and
Definitely.
quantitate what's on our EVs. So thanks for your contribution here. And thanks all for being here today, and for everybody who's asked questions and for all of those who have joined. We will look forward to seeing you again on another EV Club in the near term. And if anybody has a exciting paper that they've seen out there that they would like to have featured on here, please feel free to reach out to me and suggest it. So have a great rest of the week, everybody, and good luck with your research. And thank you again. Bye now.